Well guys, today I'm a registered pharmacist. As a pharmacist, it is my job to dispense medicine suitable for the ailments or illnesses of my customers so that they'll have a better health and or a better quality of life. That's why it is my duty to know the workings of drug production and have expertise in the safe use of prescriptions beforehand. Day-to-day, I encounter, study, and relate medicines to pharmaceutical biotechnology. Pharmaceutical biotechnology employs living things or parts of living things in order to make pharmaceutical and diagnostic products, in short, medicines. For example, the serendipity of penicillin. Penicillin was accidentally discovered in 1928 by Dr. Alexander Fleming, a Scottish physician and microbiologist. It was discovered in a moldy Petri dish in St. Mary's Hospital, London. After returning from a holiday, Fleming discovered mold growing on a Petri dish of Staphylococcus bacteria, known for causing various blood infections like abscesses, boils, furuncles, and cellulitis that could eventually lead to sepsis or death. However, the observed developing mold appeared to be stopping the bacteria from growing. Thus, he soon identified that the mold produces self-defense chemical that could kill bacteria. Later today, the mold was named penicillium. How exactly does the penicillium kill the bacteria? Well, when bacteria multiplies, they develop little holes in their cell walls. To fill these holes, it is the responsibility of the peptidoglycans to close them out and re-strengthen their cell walls. And it is at this time the presence of penicillium get to kill the bacteria, as penicillin prevents the peptidoglycans to do their job. The holes remain, and the bacteria are left vulnerable for fluid to enter the bacterium cell until it bursts and destroys it. This discovery and process paved the way for the advancement of antibiotics throughout the following years. Up to this day, the first wonder drug penicillin is still being used in the treatment of throat infections, meningitis, syphilis, and other various infections caused by bacteria. Since its discovery, following the development of a good number of antibiotics to treat various types of infections, the challenge is the rise and development of drug-resistant infections that can no longer be treated by existing antibiotics, as the continuous development and invention of new ones are non-stop.
Greetings to all of you. I am Azikir Fernandez and I'm a rhizosphere agricultural microbiologist. And as a rhizosphere agricultural microbiologist, it is my job to investigate how microbes and plants in their collection of nutrients and how they participate or indirectly participate in the biocontrol of plant pathogens. Bacterial communities in the rhizosphere are diverse with different compositions depending on the root zone, plant species, plant phenological phase, stress, and disease events. Rhizospheres support and protect plant growth, agricultural production, human and animal health by nurturing diverse organisms that are useful in biogeochemical and physical processes that promote soil health. The following techniques are used in rhizosphere research. In the study of bacterial communities in the rhizosphere, PCR-based fingerprinting techniques such as terminal restriction fragments, length polymorphism, denaturing gradient gel electrophoresis, and single-strand conformation polymorphism have been widely used. The quantitative polymerase change reaction is a molecular technique that is widely used for detecting and quantifying specific genes and their expressions in DNA and RNA samples from various environments. It is a sensitive technique that detects the signal produced by DNA fragments amplifications in real time during PCR cycle. Microarray analysis in the rhizosphere strain transcription profiles can provide information on gene expressions involved in the synthesis of several signals that control bacterial activity in the rhizosphere. GeneKeep microarray analysis of transcript profiles related to motility of Pseudomonas aeruginosus, a ubiquitous gram-negativity bacterium, is one example. Not only is the rhizosphere an important part of the plant ecosystem, but it also governs in the chemistry of plant nutrition acquisitions. plant secretes exudus such as sugar, organic acids, and amino acids, which influence soil microbial communities and activities, leading to an increased number of microorganisms. These microbes either help in plants acquire nutrients from soil or participate in the biocontrol of plants' pathogens, either directly or indirectly. Rhizosphere bacteria contribute to biogeochemical cycling of macronutrients and micronutrients, as well as determining their bioavailability to plants and microbes. So, as a summary of our lesson, rhizosphere has a huge impact on ecosystem of plant life, and it is needed for all species. That is all about rhizospheres. Now let's move on to the next topic.
Well, I am an industrial microbiologist. Haha, yes, that is lame, but it isn't really. Industrial microbiology is a branch of chemical engineering that applies chemical engineering principles to microbial processes, designing fermenters or bioreactors, and developing microbial processes for the production of enzymes, biochemicals, biopharmaceuticals, and fertilizers are examples of bioprocess engineering. It also includes genetic engineering approaches for increasing microbial product yield and quality. Bioprocess engineering is proving to be extremely useful in today's world. Although it continues to influence diverse industrial fields of activity, including food, feed, and other commodities, for example, polymer manufacture, biofuels and energy production, providing services such as environmental protection, and the development and production of many of the most effective drugs. The understanding of biology down to the molecular level opens the way to create novel products and efficient environmentally acceptable methods for their production. Several decades later, Butler disproved the hypothesis that processes in living cells required a metaphysical vis brutalis in addition to pure chemical laws. Enzymes were shown to be the chemical basis of bioconversions. Studies on the formation of products in microbial fermentations resulted in the manufacture of citric acid and chemical components required for explosives, particularly in wartime, acetone and butanol, and further products through fermentation. The requirements for penicillin during the Second World War led to the industrial manufacture of penicillin and to the era of antibiotics, with further antibiotics like streptomycin becoming available. This was followed by a new class of high-value added products, mainly secondary metabolites. Examples are steroids obtained by biotransformation. Early biotechnology had its roots in fascinating discoveries, such as yeast as living matter being responsible for the fermentation of beer and wine. Serious controversies arose between vitalists and chemists, resulting in the reversal of theories and paradigms, but prompting continuing research and progress. Pasteur's work led to the establishment of the science of microbiology by developing pure monoculture in sterile medium, and together with the work of Robert Koch, to the recognition that a single pathogenic organism is a causative agent for a particular disease. Industrial microbiology is the use of microbes in huge quantities to produce food or industrial goods. In industrial microbiology, a wide variety of microorganisms are utilized, including naturally occurring species, laboratory-chosen mutants, and even genetically engineered organisms, or the GMOs. The debate over the use of the genetically modified organisms, or the GMOs, in food sources is currently gaining traction, with more and more supporters on both sides. However, the industrial use of microorganisms is deeply ingrained in today's society.